Good evening, everyone. My name is Dr. Shannon Datweiler, and as the chair of the Department of Biological Sciences, I want to welcome you here at Sacramento State University to our Topics in Regenerative Medicine seminar series. The audience includes students, faculty, and professional and community members at large, and is intended to provide information to the general public on advances in stem cell and regenerative medical research. The Regenerative Lecture Series is co-sponsored by the Center for STEM Excellence, which promotes the advancement of science, technology, engineering, and mathematics disciplines, and the California Institute for Regenerative Medicine, or CIRM. We are very excited to announce that our Cohort 7 students will graduate this coming May, and Cohort 8 is progressing through the academic phase and selecting UC Davis laboratory mentors. Students in the Professional Stem Cell Master's Program, may I ask you to stand at this point? Thank you. We thank the UC Davis research scientists who trained the graduate students on stem cell therapy. If you are interested in our program, please contact Dr. Rosalie Sprouse for additional information or take a program announcement located on the side table. Our past, current, and future speakers' seminars can be viewed on the Sac State STEM website. Please take a moment to silence your cell phones. We will have a brief question period at the end of the lecture. Tonight, Dr. Fink will present his talk on transcriptional regulation of genes implicated in neurological disorders. Kyle Fink has a PhD in neuroscience from Central Michigan University and a PhD in neuroimmunology from the University of Nantes, France. He is currently at UC Davis Medical Center, where his research team investigates genetically linked neurological disorders such as Huntington's disease. The aim is to develop a therapeutic pipeline in which precision medicine can be used to identify disease-causing genetic variants to advance neurotherapeutics and translational medicine. Specifically, how to deliver these therapies to maximize biodistribution while minimizing the immune response to increase safety and the therapeutic potential of this approach. He is a member of several professional organizations, well-published, and his work has helped to advance a gene-modified stem cell therapy to pre-IND meeting with the FDA in order to begin a phase 1 clinical trial for Huntington's disease. We are fortunate to have all of you here with us this evening. Please join me in welcoming our honored guest, Dr. Kyle Fink.
Thank you. Thank you for that very kind introduction, and thank you again for the invitation to be able to speak here tonight regarding some of the work that's being done in our laboratory over at the Davis Medical Center here in Sacramento. And so the title of our talk, again today we're going to go over several different aspects of what a lot of these different words mean in terms of transcriptional regulation, some of these genes that may be implicated in neurological disorders, and then how we can kind of use the current landscape and technology to be able to use or combine these two in what we hope is a novel therapeutic application. And so again, I'm at the UC Davis Stem Cell Program, which is at our Institute for Regenerative Cures. We can see this is our building here, just off of Stockton Boulevard. And so I'm going to spend the first couple of slides just kind of showing you guys some of the different things that our program does and some of the different opportunities and research that's going on in our center. I'm just a very small piece of a very dynamic research team that's present. And so really, we think of ourselves as kind of the center of the universe in terms of our stem cell program and with all the different diseases and all the different things that we work on. And so we can see coming around our stem cell program here are all the different disease indications that our team and our lab is currently working on. I'm up here on one of those top bubbles working mostly in Huntington's disease, juvenile Huntington's disease, and other neurological disorders that are genetically linked. When we take a little bit more global look of what we do within UC Davis, we have several different departments that we work with. We work very closely with our veterinary school, actually trying to develop therapeutics for what we consider compassion animals that also get spontaneous diseases, as well as like the neurology department and our Huntington's Disease Center of Excellence, which is an amazing clinic that follows over 400 patients of Huntington's disease. And then with this as well, we also try to follow these 4 pillars. And so the leadership of UC Davis, following the scope of research that we want to pursue, which is advancing these novel therapeutics for unmet medical needs, we do work a lot with industry partners of how we actually can advance these therapeutics towards a clinical trial. And then the main reason I'm here today is community and education. So I'm hoping for some of the students in the room and some of those lifelong learners, we'll be able to provide a little bit of education of where we are currently in the field. And then in terms of community, because you guys are all part of our Sacramento community, you guys are all citizens of California, you all live here, and you'll really help get to decide how we shape this research moving forward. And so a little bit about what we're going to talk about today. We're going to talk a little bit about gene regulation. So we're going to go back to a very basic view starting at the DNA level of how we think about genes. I'm going to tell you a little bit about artificial transcription factors and how we can use these in a therapeutic manner. These are really cool things like zinc fingers, transcription activator-like effectors, or the CRISPR-Cas9 system, which gets a lot of press these days. And then we'll apply them to a neurologically linked disorder and talk about how we think about therapeutic options approaching this, how we can use traditional gene therapy, stem cell therapeutics, and as well as these novel approaches. And then we'll spend a little time at the very end kind of talking about where we think this research is going and what we think are some of the unanswered questions and unmet needs that we need to address before we really move this forward. And so go back to stem cells. And so when we really think about our bodies, they're very, very complex. We have about 220 unique cell types that make up our body. And so these 220 mature functioning cells are skin cells, hair cells, teeth, blood. They all come from stem cells. They all differentiate from a single starting cell source. And what that looks like when we think about a stem cell is basically a cell that has the ability to self-renew. And outside of being able to self-renew, these stem cells are little cute yellow cells here that are dividing, and one cell is becoming 2 cells. And so about after 20 of these population doublings, we'll have over 1 million cells, and this continues to grow, and we make new cells every day. They also have the ability to differentiate, and this is what really makes a stem cell a stem cell. And so this little yellow stem cell that we have here is differentiating now into our nice little green square, and this is our progenitor cell. And what this happens to be is a skin progenitor cell. So still starting from our same yellow stem cell, we now have a skin progenitor cell that's eventually going to differentiate into a functional skin cell. But the same yellow cell is continuing to divide, but in this case, it's making a little pink triangle cell, which happens to be a neural progenitor cell. And it's a neural progenitor cell because I'm a neuroscientist and I really like neurons, and so everything is always going to relate back to the brain. And so from the same starting cell source, we have this yellow cell that's now given rise to a skin cell, a functioning skin cell, as well as a functional brain cell. And in this video from the Howard Hughes Medical Institute, we can kind of see what this development looks like. And so again, starting from these single cells, this one cell source now, it's growing and dividing into multiple cells. And now we have this nice little cell, and when we look at this inner cell mass, everything in the human body is going to come from this single set of cells. And these little cells that were just highlighted here, these are going to be our blastocytes. These are 8 cells that are going to basically differentiate into everything needed for complete embryogenesis. And these different cells, as they continue to grow and they divide, they're going to start to get more specialized. And as they start to get more specialized, these cells are going to differentiate into different regions. Our ectoderm, which is again going to be the one that I like, is going to make all of our central nervous system, all our peripheral nerves, as well as this amazing organ we have called the brain, as well as our skin cells. We also have the mesoderm, and this is what's going to create all of our smooth muscle, as well as our vascular system, our veins, and different organs. And then lastly, we have our endoderm, which is going to constitute all of our internal organs, our intestines, stomach. And so what's really interesting is starting from these single cells, these identical cells that we have inside of our body, we give rise to the entire human body. And so again, if we kind of look at this, we have our initial cells, our fertilized egg or blastocyte, goes through embryogenesis, and then we end up over here, these 220 different types of cells. So how do cells know what to do? How do these identical cells that started from these 8 initial cells, how do they develop? How do they take on these very specialized roles? And so if we look closer inside each cell, we have a nucleus, and then we have a chromosome, and we have the DNA here. And this DNA is basically the building blocks. These are the recipes that tell every cell what to do. And this is going to be identical in every cell. And so in our human genome, we have 3.3 billion base pairs. These 3.3 billion base pairs are stored across our 22 autosomal paired chromosomes, and then the last two are going to be these sex-linked chromosomes down here. If you have two X's, female, X and a Y for male. These different base pairs are going to code for about 20 to 25,000 genes, and we've mapped this fairly completely in the human genome, so we kind of know what genes are there and what this gene function is. So approximately 1.5% of all of these base pairs actually code for a protein, and this protein is going to cause these specific cell functions. And so what you can see over here in the video, this is our DNA, these double helixes, and they started forming our nucleosomes. Now we've formed a chromatin fiber, and they continue to condense and tightly wrap around in these very distinct structures. And so we see as this fiber continues to grow and get more complex and keep wrapping these 3.3 billion base pairs, basically the recipe of every cell function that we have in our body, eventually they're going to be stored in our chromosomes. And so these are a lot more of the familiar shapes that we know. And here we see a dividing cell in which you can actually see two sets of the chromosome up on the top and on the bottom here. So again, when we think about all of this DNA, all of these different base pairs that we again have our traditional, what we think of the chromosome, our chromatin structure coming out into our nucleosomes around these histones here and winding down to our single DNA double helix and our base pairs. And these base pairs are consistent of these four building blocks. And so every cell has identical DNA. Every cell encodes all 46 of these chromosomes, the pairs and the sex-determining chromosomes. But how does the cell know what recipe to read, what function to do? And so this is controlled by regulatory domains and gene transcription. And so this is our normal, what we think of as a gene and a promoter. Within our DNA, we have our polymerase land, and it makes our gene product. Those little squiggly lines that we saw coming off of the side here was our RNA transcript. This is eventually going to be coded into protein. And what's very interesting is the body doesn't only do this based off the gene and the promoter, but off of these regulatory domains, enhancer regions. And so here we can see down here, within these switches, we have a regulatory molecule. And what this regulatory molecule is going to do is sit just upstream of the promoter and actually recruit this polymerase to drive gene transcription. There's other molecules that we can have that are going to land at other distal regions within or near the gene, and they may be regulatory molecules. And what these regulatory molecules may do is just help in recruiting extra polymerases, modifying how this gene is expressed. And so during development, different cells, as they get specialized, different parts of the chromatin, different parts of the DNA are going to be open or closed, and this is going to basically tell that cell what it should do. And so if we go back to our central dogma again, we have our cell here, we have our DNA, we have the RNA being made, RNA messenger, RNA here. It gets made into protein, and this is going to cause the different specialized functions of all of our different cell types. And this is basically caused by which regions of that genome are going to be read. So you have all of these base pairs, you have all of these copies in every cell of your body. But how do they actually determine which ones are going to be read? And this is a major question that we try to address and try to understand. So again, we're going to come back to this central dogma several times, from DNA to RNA to protein. So we have these 3.3 billion base pairs, And so what happens if something goes wrong? What happens if these base pairs are deleted? What if they shift or they change? And so down here we can think of inherited mutations here. So within these initial starting cells, we may have a genetic mutation that's occurring. We could have genetic mutations that happen during development, things like autism, development that's going to occur here, or even once the cells are mature and fully functional, we could have mutations that are going to happen again. And this could be things like cancer. And so we're going to spend a lot of time talking about a specific genetic disorder called Huntington's disease. And we're going to use this as our model disorder in which we can use the knowledge that we have around DNA transcription and translation to develop novel therapeutic approaches. So in Huntington's disease, for those who aren't familiar with it, here on chromosome 4, exon 1, we have these trinucleotide CAG repeats. And up here in the top, we can see this is a healthy individual that has less than the threshold that would cause Huntington's disease. It's going to make a normal protein, and this normal protein is going to result in healthy, functional neurons. In the case of Huntington's disease here, we have an expansion of these CAG repeats, and we typically set the threshold of greater than 38. So if you have greater than 38 CAG repeats, this is a dominant disorder, meaning you only have to have one copy of this gene, and it's going to result in this gain-of-function mutant protein, which eventually is going to lead to neuronal degeneration. And so here we have a bunch of cells talking to each other. And these cells are undergoing regular transcription. They're relying on proteins and protein synthesis to do their normal function. They talk to each other, and they spread messages throughout the brain. So what happens in Huntington's disease is these cells are not going to be functioning normally. They're going to have an abnormal protein that's going to cause a slight dysfunction. This could be in cortical reorganization during development, as well as several other different things that the mutant protein can actually do. And so this very specific region of the brain that's most affected is what we'll collectively refer to as our basal ganglia structures, our caudate and our putamen. And so in these MRI scans that we have here, anything in green, this is something that's normal size, as related back to a population control, age-matched, sex-matched controls. And then over time, what we see on these coronal views, so looking through the brain in this plane, we can see anything in red means an enlargement, anything in blue means atrophy or a shrinkage, cell loss, or a reorganization. And so we can see over 18 months in these early stage Huntington's disease patients, we see significant dysfunction, significant loss of cells. Here in our axial plane, which is going to be looking through the brain in this plane, we can see this same area, the head of the caudate and the putamen here, are also showing this atrophy. And so really when we look at this, this is a devastating disease currently with no approved treatments outside of palliative care and suppressing what behavioral abnormalities will manifest with it. So when we really approach any type of neurological disease, at least in my view, we kind of approach it with three main treatment options that I kind of go to first, and this is mostly due to my training and the work that we've done over my career. And the first one is going to be neuroprotection. And so neuroprotection would basically mean let's go in there and try to prevent whatever dysfunction is happening in these cells before it starts to occur, before it starts to happen. In the case of Huntington's disease, it may be adding a growth factor that we know is downregulated from this gene. We could also think of cell replacement. And so now we have the technology to make induced pluripotent stem cells. We can make them into neuronal-like cells. And we could potentially transplant them into the region of where the cell loss occurs. And then the last one, with the new technologies and the technological advances that have come on for researchers and scientists in the last about 6 or 7 years, is being able to actually go in and look at this disease at the genomic level and see if we can make a genomic modification or correction. In the case of Huntington's, actually trying to suppress the expression of this mutant gene. This is mostly what my lab works on, and this very handsome fellow who's sitting in the front row tonight, Peter, does a lot of this work with me. So I'd just like to acknowledge him early on in the talk. He's in the thank you slides at the end too, but a lot of this work has really been driven by him and really has helped develop this and take this to the point that it is now. And so when we think of different gene therapies, some of the different things that we think of, at least traditional gene therapy, is we isolate a gene that we know may be lacking. And then we can take this gene and we can isolate it. We can isolate the actual DNA copies of these, the base pairs, the recipe for whatever protein or gene may be lacking. We can encapsulate it in different viral vectors, so modified viruses that we know can integrate inside of the cell and cause transcription. So basically, like we saw that cell transcribing a gene, we can insert genes exogenously into the human that will start making the protein or the factors that we need. We can also think of gene therapy now in terms of iPSCs. So we can add genes to these fully differentiated mature cells, turn them back into those embryonic-like cells, so cells that we kind of saw in that video at the beginning of the blastocyte. And then we can differentiate those into cells and then maybe bring those back and transplant them into the patient. And then really the way that I like to think about gene therapy now is we can actually go into the genetics, we can actually go into the DNA and try to modify it. And this could be doing something like cutting out extra base pairs or causing a deletion in the genome. Or one of the things that we're a lot more interested in is actually going in and being able to modify which genes are going to be transcribed, which genes are going to be expressed. And genes that are maybe on, that we don't want on, we may be able to turn off. Genes that may be silenced, we may be able to reactivate. And so I'm just going to kind of go through a couple of schematics of what we think about in practice for these different approaches. And so in neuroprotection, we see this very complex cell down at the bottom. with all of these different things. I don't expect you to read or understand any of these things. This is a cell that lives in the striatum, or the basal ganglia of a Huntington's disease patient. This is what we call a medium spiny neuron, and during the course of the disease, in the presence of this mutant gene and the mutant protein, the cell is eventually going to start to dysfunction, and it's going to undergo cell death. And this is what we saw in those MRI pictures. This blue area of the brain, these cells are dying off. So what we would like to do before the cell ever starts to die is we'd like to try to give it a little boost. We'd like to throw it a life raft and keep it alive and functioning and a little bit happy a little bit longer, and hopefully delay the disease progression. So we propose to do that by transplanting an adult stem cell called a mesenchymal stem cell, isolated from the bone marrow, which are our beautiful little blue or green cells up here. And then we genetically engineer them to release the factor that's lacking, called brain-derived neurotrophic factor, or BDNF, or these little red dots here. And so hopefully these cells can get to the neurons, secrete BDNF. This will go to the cells and keep them alive and functioning a little bit longer. So we've been doing this work for a while, where we transplant these cells into animal models of disease. We see that they can secrete BDNF, that they can engraft inside of the diseased brain and cause a little bit of functional recovery. It depends a lot about how we get these cells, what source we get the cells from adult different populations, adipose tissue, bone marrow. We can get them from umbilical cord. How we maintain them in culture greatly modifies how they function once we transplant them. We see that when we put them into models that exhibit some of the phenotypic characterizations, like a motor deficit, the animals that receive the cell transplantation perform a little bit better. This is the study that we've actually gone towards with the FDA to have a pre-IND and hopefully be submitting an IND to take this forward with a phase 1 clinical trial. This is a manuscript over those large studies. And what we can see here is behaviorally, we have our healthy mice, the mice that don't have Huntington's disease here, and this is just measuring exploration in this box over a certain time period. And we can see that most mice like to hang out in the corners, as indicated by these warmer red colors here. But we see less activity indicated by the blue and the darker colors. But these wild-type mice are still exploring. They're still going in the center of the box. When we look at a mouse that has Huntington's disease, we can see that this area is almost devoid of all activity. These mice have severe anxiety, which correlates a little bit with the disease phenotype. And we see that they just spend the majority or almost all of their time running around the edges of the box. So when we transplant these mice, actually with a genetically engineered mesenchymal stem cell, we can see that this group here are mice that have Huntington's disease that receive the cell transplantation look a lot closer to this group than they do to this group. And then when we look in their brains, what we can look at, so again, this is a quantification of those same type of images that we had before, those MRIs that I showed you from the patient. We can see our healthy mice here have a nice big striatum. Our mice that have Huntington's disease have a significantly reduced volume. But then depending on the type of cells that we transplant and the number of cells, we can see that this group resembles this group a lot closer than this group. And so we think this has a lot of potential and a lot of promise. And we're very excited about trying to move this forward and actually start a phase 1 clinical trial. So when we think about cell replacement, what happens if the cell is already gone? So what if we didn't get there in time to actually do neuroprotection and rescue the cell? So is there a way in which we can place a cell in here that's going to do the same thing? And the answer is kinda. So we can make an induced pluripotent stem cell, which is a cell that's we can push through that normal development. It's going to be embryonic-like, and then we can put it into a neuron. And then we can transplant it into the area that the cells are getting lost, so that blue area that we showed you earlier. And I promise you guys that I actually put a lot of effort into this talk, and this isn't just really poor Photoshop skills. So I can ask you guys, are these colors of the cells, are they the same? No, not quite, but they're close. And if we remember that diagram that we saw, the inside of this cell had all kinds of arrows and different kinds of complexity and different kinds of things inside of it. This one doesn't. It has some of the things. It has our nucleus here, it has some nuclear pores, this little squiggly lines of mitochondria. So it's kind of like the cell that was lost, but it's not exactly the same. Then when we actually look here, let's look at the connections that this cell is making. We can see that this one lines up. It's pretty good, a little off down here, and then this one it's not quite the same. So when we really think about cell replacement, I think this is about the current state of where we are within the scientific development and the scientific pursuit. We can make a cell that kind of looks and kind of functions like a neuron. We can transplant it in the area that it's needed. It's going to kind of function the same way, and it's going to kind of connect the same way. And we've done a lot of this work. And so we've transplanted these cells into rat models of Huntington's disease. this picture up here where we can see this little crosshair, this is a transplanted cell that's kind of looking like the neuron that we want it to be, this medium spiny neuron. When we transplant it into an animal and we look at the functionality, what we can see is over here, the cells are kind of becoming what they should be. This blue line indicates our healthy rats, the rats that don't have Huntington's. Our red line here is the animals that are progressing and getting significant more motoric dysfunction. And then this green line here, this is an animal that was treated. So we can see that we can rescue some of these phenotypes. We can kind of help make the animals a little bit better, but we're not fully recapturing the deficits that were observed. And Peter and I have written a lot about this. We've done a lot of work. So we published this paper. It has a huge table in it with all the different work, not only from my group, but other groups around the world. We've done this for what it would look like for a clinical trial for adults and kids with Huntington's disease. And then we've also really honed in on this function of BDNF and how we think BDNF could be harnessed and leveraged for neurological diseases. These are all really cool papers that we've recently published in the last about year and a half. So let's really move into what I think is very cool and very interesting. So what about if we look at the genetics of Huntington's disease? So again, I showed you guys, here's our cell again. But now, I'm telling you that this little altered gene down here is what's causing the cell death. So this is the guy that's causing all this downstream cellular dysfunction, all these things that we see around the outside. And it's with this little mutant Huntington protein. And so is there a way in which we can actually target this and prevent any of those other dysfunctions from happening? And so when we think about what the targets are, we know that the mutant protein is going to cause some of this dysfunction. So there's ways in which you can target the protein. We could try to overexpress something that's going to degrade the protein. We could try to knock down the protein itself. And then when we think about that central dogma, we could also think about targeting the RNA. And there's a lot of techniques and technology that's been available to actually target the RNA. But really what we were interested in is targeting directly the cause of Huntington's disease. So when we think about this in a schematic, so with this central dogma, again, you guys have to entertain me. We can think of the protein as the leaves of a tree. The RNA is going to be the branches of the tree, and then the DNA is down here. This is our roots of the tree. And so what's going to happen if the leaves fall off of a tree? You take the leaves off of a tree. Well, after some time, the leaves are eventually going to grow back. What happens if we could take the branches off of the tree? What if we go cut the branches off of the tree? Well, again, it's going to prevent the leaves. It's going to prevent this protein from being formed temporarily. But after some time, the branches are going to grow back and the leaves are going to come back on the tree. So what happens if we actually go and we take the roots out of the tree? What if we kill the roots of the tree? There's going to be no more branches and there's going to be no more leaves. And so this is really what we want to try to do. And so how we do this is we're trying to harness something within the body that naturally occurs already. And this is using transcription factors. And so what we can think about here, this is our central dogma again. This little complex is forming in the promoter. We have our gene. When this receives the correct signatures and the correct signals of the cell, we have these enhancer regions or an activator. This is going to land, and it's going to regulate the promoter of the gene, and it's going to start transcribing and making our RNA transcript. And so what we have is we have multiple regions within the genome, and we showed this a little bit earlier with these switches and these regulatory domains. So we have these enhancer regions as well. And so here we have a mediator. And so what this mediator is going to do is it's going to recruit this mediating protein, and it's going to cause the polymerase to then transcribe the gene. But we also have transcription factors that function as suppressors. And so this was the red dot that we had earlier, and we didn't talk about that for a reason. We wanted to save that for now. And so what happens when these repressors land, they can land in the enhancer region, and they can land in these different regulatory domains. And basically what they're going to do is they're going to block transcription. And so even if the mediators are being recruited to the correct spot, a good repressor will be able to stop transcription from occurring. And so this is a way that our body naturally regulates gene expression throughout life and throughout development. And so some of these that we're very interested in, if we remember those really tightly coiled DNA loops earlier, they can actually cause chromatin remodeling. And what's going to happen on this chromatin remodeling is this normally open gene is now going to be tightly condensed and not allow no binding of anything. But it's also potentially reversible. So even if you have a gene that's tightly compacted and has these, this chromatin remodeling, we may be able to land an activator there that's going to relax the chromatin and again allow for gene transcription. But we really like these artificial these transcription factors that naturally occur in the body for being able to silence gene expression. And so what you may think is, well, that's really cool. You can bind these around and they seem to go to different promoters. And so this is how the body's going to naturally regulate genes. But how do you get this promoter to the right spot? What if it's a gene that the body thinks should be transcribed, but we want to be able to turn off? How do we get this really cool suppressing agent that's going to shut off gene production to the correct spot? And the answer is we use these really cool molecules called transcription activator-like effectors, or we harness the CRISPR-Cas9 system. And so what these two modalities are, I'm really partial to the talls. I'm warming up to CRISPR because Peter's making me. But what this is this actually recognizes base pairs. And this is part of a biological innate immunity. And so what this does is it actually recognizes DNA very specifically. So these base pairs that we have, we can synthetically make these to go and find that sequence and then tightly wrap around and bind to it. And then what we have is these little spaces on the end where we're able to put an effector domain. And these effector domains can be these transcription factors that are going to change the regulation of genes. And so here, this is our friendly little transcription activator-like effector, or a TAL, bouncing around the genome, recognizing the sequence that we like, and tightly wrapping itself around the DNA. Over here, the CRISPR system is actually this little red loop. This is our guide RNA, and this big, cool-looking guy around the outside, this is our Cas9 protein. And so what happens here is this guide RNA recognizes the base pairs, it binds, recruits Cas9, and then we have an effect. And what we actually see, this little bright light that happens here, this is a double-stranded break and a deletion. And so what we can do with these is we can rapidly synthesize either one of these targeting modalities to hit any base pair sequence we want within the genome. And so if we have a signature of a disease or a gene, we can design these to specifically land in those spots and then make a functional change. They're highly efficient, and they're very highly specific if they're designed the correct ways. And we think they have minimal off-target effects, but this is something that we're currently evaluating, and we're constantly evaluating along with everybody else in the field. And then this last part, I think, is one of the cooler parts. We're not really that interested in just actually creating a double-stranded break. What we're actually doing is very interested in being able to change transcription and change how those genes are expressed. And we do that with our many different flavors of gene regulators. And so these different effector domains that we can attach onto each one of these targeting modalities, we have activators that can recruit transcription factors or drive transcription in and of themselves. We also have things that can cause methylation, so causing that region of the genome not to be open for transcription anymore. So these ones can open those regions to allow transcription. These ones can drive transcription on their own. We also have repressors that can cause chromatin remodeling, or methylation to close off those regions. Again, I really like transcription activator-like effectors. They're kind of the middle child of genome engineering. Zinc fingers have been around for a really long time. They're kind of difficult to work with, hard to synthesize, hard to understand. But if you're able to harness their power efficiently, they're a very, very powerful tool. That's why they have a not very cool cartoon over here on the left. Our transcription activator-like effectors, again, are middle child of the genome engineering world. Kind of get a little bit cooler cartoon, but shortly after these guys were discovered, the CRISPR-Cas9 phenomenon hit, and then enough labs had graphic arts money to be able to design these really cool things. So I apologize, but this guy really here in the middle is very cool as well. And zinc fingers too. They just haven't gotten their just due. But I think as we move forward, we're really going to be able to pick through these different DNA targeting modalities to choose which one would fit the disease in the most efficient way. And so when we actually put this in practice, what we've been able to do is isolate skin cells, human fibroblasts from patients that have Huntington's disease. And they have different, these are different ages, they're different sexes. We maintain these cells, they have different numbers of repeats. But what we're able to do is they all have the same, or these top rows at least, have the same DNA signature in the mutant allele. So what these red bars are, and what this will be for the rest of the talk, this is the expression of the mutant gene of Huntington's disease. In blue, this is our healthy gene. And so what we can see here, if we have this very specific signature, and this is a transcription activator-like effector paired with a CRAB repressive domain, which should cause that chromatin remodeling, we can see that they all result in a significant reduction of the mutant gene without really affecting the expression of the healthy wild-type gene. If they don't have this very specific allele signature, we can see that there's no effect. And in some rare cases, and we've been able to find and isolate cells that have this really unique diseased allele signature on both the disease and the healthy gene, we see knockdown of both. So what we think this means is we have a very highly specific, potent modality to be able to turn off the Huntington gene in human Huntington fibroblasts. And again, this is done in cell culture. So then we wanted to test it in neurons. And so what we did was we isolated neurons from a mouse that has Huntington's disease. We see these pictures up here. In this case, we made our transcription activator-like effector with crab have a little fluorescent protein on the end of it that would turn all the cells that get treated red. And so we can see in both the striatum, so this is that area that we talked a lot about, and the cortex. So when we isolate neurons from the striatum and the cortex and we culture them, we can see that our healthy wild-type neurons don't have, or have very little expression of any mutant gene and the wild-type gene. Our transgenic animals that don't get treated have high expression of the mutant gene, but when we treat them with our transcription activator-like effector, we see significant reduction of the mutant allele. We've also just expanded and started doing this with the CRISPR-Cas9 guided system, and so this is using a Cas9 protein that would be nuclease deficient, so it's actually not going to be able to make a cut. So we call this dead Cas9 or dCas9. And then we use our different flavors down here of the different epigenetic silencers. So FOG1, crab is what we just showed with the transcription activator-like effectors, and DMNT3A, which causes a methylation mark to occur. And what we see in our human Huntington fibroblasts, again, compared to our untreated cell type, we see significant reduction of the mutant gene without affecting the expression of the healthy non-expanded allele. And so one of the major questions that we have is, how do we actually get this? So this is really cool. We've been doing it in culture. We think it's really exciting. But how do we actually get this to the place that it's needed to be? I think if we told a Huntington patient that we could cure their skin cells from having Huntington's disease, they probably wouldn't be that interested in that approach. So we need to be able to find a way to actually be able to get this into the brain. And so what we've been doing is we're working with different collaborators, trying these different delivery modalities, and one of them is using a lipid nanoparticle. So this is a company up in Vancouver called Precision Nanosystems. They use microfluidics to actually encapsulate our therapeutic plasmid into these lipid nanoparticles. And these lipid nanoparticles have cholesterol around the outside. Inside they have these ionized things that will bind, and you can see the little DNA plasmids inside. And what actually happens is this can help transport. When we place these little nanoparticles inside of the brain of a mouse, it actually allows our plasmid to get inside of the neuron and then express. And so we've been doing this, and this is a mouse brain that has Huntington's disease, and we did a unilateral transplantation. So you can see here, this is where we actually transplanted our lipid nanoparticle. So the lipid nanoparticle itself has a red hue to it. Our transcription activator-like effector in this case was green. So anywhere where we see these oranges or yellows, this is where the actual therapeutic is making it into the brain. And so we can see we actually have widespread biodistribution across this area that's affected by Huntington's disease, as long as it's moving along, as well as it moving along the white matter tracts of the corpus callosum into the subventricular zone where adult neurogenesis happens, and really spreading throughout. And so interestingly enough, we were very excited. This was a very unexpected finding for us. We didn't think it was going to work this well this early. And when we actually started looking at higher magnification pictures, so this is again one of those coronal sections looking through the mouse brain this way, we were really shocked at how much coverage we had from a single injection. And we started looking at high magnification. What we were actually able to see is in the cortex, and these are different forms of the lipid nanoparticle, and in the striatum, so this area that we're very interested in, so again, we're looking for the green dots. This is our actual therapeutic plasmid. We're able to see that it's getting into the cortex, it's getting into the striatum, and when we looked at high magnification, actually on the contralateral side, so the opposite side of where we transplanted, we were able to find this therapeutic plasmid again, creating this therapeutic protein. And interestingly, when we actually look at the gene expression again, we can see using specific formulations, we can see a significant knockdown of the mutant gene, both in the striatum and in the cortex, depending on the different modalities that we use. So what we're planning on doing moving forward is actually using these techniques to test and look at functional recovery. So we have a mouse model that actually has the full human sequence inside. We can see in blue here, these are our healthy mice again. In red, these are the mice that actually have the mutant gene, the mutant Huntington being expressed. We can see this is a measure of motor coordination called the acceleron, and we see this over time as the animals age. The Huntington's mice get significantly worse. We looked at an image like this earlier from some of those studies that we delineated with the cell transplantations. This is quantifying the volume of that structure that we're very interested in, and we can see that this is significantly reduced. So what we hope is if we're able to transplant or inject our animals early enough in this disease progression that we won't see this same functional deficit or this cell loss. These high magnification images down here we can see, I don't know how clearly you guys can see it out here. This is a mouse that has Huntington's disease. We see that it has a lot of the aggregates, the mutant protein actually forming within the brain. Our healthy animal doesn't have any of these. And so what our planned studies would be is evaluating and seeing if we are actually able to shut down expression of the mutant protein and the mutant gene in a significant way, that hopefully this deficit wouldn't occur, this deficit wouldn't occur, and our diseased brains would actually look a lot more like the healthy brains. And so how do we apply this? So Huntington's disease is a really good model disease. It's a disease that I'm very passionate about studying. I've been studying for almost a decade now a specific therapeutic development for Huntington's disease. What are other diseases that we can apply these same techniques and these same technologies for? And so we could do gene activation studies. So like what we said is we can basically target any sequence of the genome and look at any gene. And so if there's a loss of function, so a gene that should be transcribed and that may be silenced during imprinting and during development, if there's a gene that's just been mutated and causing a deficit because it needs to be present, we may be able to actually go in and reactivate that gene. And so in these loss of function diseases like Angelman's, which we specifically study with a wonderful mentor named Dave Siegel over at the Genome Center in Davis, we can use a zinc finger to hopefully be able to reactivate one of the genes implicated in the progression of Angelman's disease. We just recently started working on CDKL5 deficiency, which is a rare form of an infantile epilepsy that these kids start to develop almost 1,000 seizures a day around 6 months of life, and that continues for the first several years. And what they know is from genetic sequencing that there's mutations that occur in this kinase, this CDKL5, and when this is lacking or there's a low amount, that the brain's not developing normally, and it results in this infantile epilepsy. And so we're beginning studies right now using our transcription activator like effector and our CRISPR platforms to be able to recruit and find this actual gene and actually try to, it's an X-link, so it's on this sex chromosome, and trying to reactivate potentially the healthy gene. In terms of gene silencing, that's a lot what we talked about just now with Huntington's disease. So these may be gain-of-function diseases or disorders. And so in Huntington's disease, we know we have a mutant gene and that's making a mutant protein, and it's causing all of this downstream cellular dysfunction. And so really what we would like to do is be able to target that gene specifically and shut off production. There's also other genes that may be causing gain of functions, things, specific gene activations in possible forms of cancer. And so I'm kind of coming to the end of my talk now, and so I think what I want to do is maybe leave you guys with some unanswered questions from us in the research community. And some of these things that I think we would like to move forward with. And one of those is how long would these artificial transcriptions factors last in an organism, in a complete system, not just in our cell culture dishes in the lab, but when we actually put them inside of a living animal, inside of a living brain. And we have an idea about how long this lasts, and I think in a disease like Huntington's disease, it's pretty clear that we'd like to shut down production of the mutant protein forever. We'd like to get rid of this protein and this mutant gene throughout life. But when we start to think about diseases like the CDKL5, if we reactivate this gene, should this gene always be on? And should we actually have something that may be turning on and turning off as we go through development? And so the next question that we have is can we actually make permanent or unreversible changes? Can we make things that happen to the cell where we maybe cause such a great chromatin remodeling or a histone mark or something like that, that we could render a gene unactive or deactive for life, or turn on a gene? And then one of the major questions that my group specifically is very interested in is how do we actually get these to the brain? I love the brain. It's one of the best organs in the body. I'm very biased because I'm a neuroscientist, and so I like it a lot. But how do we actually get it to the brain? The brain has all kinds of really cool self-defense mechanisms. We have this amazing little bone casing called the skull, and so it's not really that accessible. And then underneath the skull, we have something called the blood-brain barrier. And the blood-brain barrier basically is going to filter off anything that's trying to infiltrate and actually get into the brain. Again, a lot of the stuff that we do, because I did a lot of transplantation work, if we need to get something to the brain, we go to the brain and we deliver that. But when we think therapeutically, a lot of patients may not opt to actually have a neurosurgical technique performed on them. And then once we get to the brain, do we want to target all the cells all the time? And so what we showed you was a very non-specific uptake of our therapeutic protein in a Huntington's brain of a mouse. And so this is getting all over the place, in the neurons, in the astrocytes, in the microglia, potentially back to the hippocampus and the cortex. And then what if this disease is really not affecting all those different regions of the brain? What if it's a very specific subtype of cells that may be affected? And is there ways in which we can target them very specifically? So we can only have our therapeutic protein present when we want and where we want. And I think the last question, and this is a question that actually got raised earlier tonight when I was talking with the stem cell students, but what genes should we target? And as we start to get more information with bioinformatics and with all these new very cool sequencing techniques, we can start to uncover gene profiles that are implicated in different types of diseases. So in Huntington's disease, I think the answer is very, very obvious. What gene should we target? Well, we should target the Huntington gene. And what should we do? We should try to turn it off. In CDKL5 during development, it's the lack of this gene and the lack of this protein is causing seizures. So what should we do is we should go and we should turn that gene on. But what about things like schizophrenia or autism spectrum disorders, where it may be 1,000 different genes that are differentially regulated? What about things in adulthood, like post-traumatic stress disorders, which may be causing epigenetic changes to your DNA and to the epigenetic landscape of your cell? Should we go in and should we try to reverse those genes? And which genes are fair to target and which genes aren't? Luckily, I have blue eyes, but would it be fair to go in and make all your children have blue eyes? Could we turn on that gene? Could we rearrange that? And that's a question that I think potentially the tools are getting us to the stage that we may have to start considering and trying to answer. And so again, I'm going to leave you guys with a little bit of what our therapeutic pipeline is and what we think of how we can approach different diseases. And so really what we do is we have our amazing human body over here, and again, this is a central nervous system, our peripheral nerves, and all those kinds of things, because I'm a neuroscientist, and that's what I'm interested in and what we like to do. And so if we identify a gene that's, or a disease or a different indication, what we can do is we can identify that. We potentially can just take a fibroblast or a blood sample from that patient, and we can culture this in the lab. We have the techniques now, and we're developing these different things that we can take them to an induced pluripotent stem cell, or maybe actually jump that stage and go right to a neuron. So we can culture neurons from a human patient that has a disease in our lab. We can use all kinds of these next generation sequencing modalities to actually get down and look at the single base pairs. And so this is actually a single nucleotide polymorphisms that exist in regulatory domain of the Huntington genome. And we love this little hash mark that goes here, because this little hash mark means that we can target this gene with our different transcription activator-like effectors and try to shut down only production of the mutant protein. And so we can look at the genome and we can design our different modalities, our CRISPR-Cas9 system, our transcription activator-like effector to specifically bind to these sequences and create a change. Then we can test that in cell models again in the lab, in our human cells that we can culture, in the neurons that we can make, in neurons that we can isolate from animal models of the disease, and then move them forward and actually test them behaviorally. We can transplant them into a mouse, we can inject them, and then we can measure all the different behavioral deficits and hopefully behavioral rescue. And what the idea with this pipeline is that we can move forward and actually develop meaningful therapeutics for those patients who are suffering from these unmet medical needs. So again, there's a lot of people that I need to thank. Jen, who directs our center, Vicki Wheelock, who runs the Huntington's Disease Center for Excellence, who's been a major supporter of this work and also helped us develop a lot of these different things. Dave Siegel, who runs the Genome Center. That's the one who gave me Peter to work with us and really helped advance this research, as well as really help mature these projects and form them into hopefully a meaningful therapeutic. Peter, who I mentioned, and Vita's an undergraduate, and then we have several volunteers. This is our most recent picture here. This is our lab. And so they all participate and help us. We've been very lucky to get funding from several wonderful foundations, as well as some national and international institutes to continue this work. So I think with that, I'd be happy to entertain any questions that you guys may have. Thank you.
So when you're using TALEN in CRISPR, would you have to design a specific targeting for each patient as a different variation of the mutation?
You could. And so a lot of what we're doing and what we have here, so this single nucleotide polymorphism that exists right now in this patient, what this specific a single base pair change actually indicates occurs in about 44% of the patient population. So this identical sequence would cover 44% of the patient population. So if you could come in and we could do sequencing, we could say, yes, you would be eligible for this tall. We have 3 different SNPs that we've designed talls for that would cover theoretically about 87.5% of the Huntington's population, and then have recently added 6 guide RNAs for the CRISPR-Cas9 system that should be able to get us closer to the mid-90s of coverage for the patient population. So it would be personalized, but it wouldn't be patient-specific necessarily.
And since these patients are already prone to, seems like, genetic errors, how do you know that these things won't target other non-known silent mutations in different areas?
And so the Huntington gene itself, the mutation, the actual expansion, is fairly stable. If it's paternally transmitted, sometimes you'll get a little bit of an elongation of what the CAG repeat number may be, and this happens during spermatogenesis. But throughout life, the number of CAG repeats you have remain fairly stable. And what's very interesting, and this comes from some different epidemiological studies, but the patients with Huntington's disease actually have cancer at a little bit lower frequency. So their genome, why they have an expansion, they have this genetic mutation, this chromosomal abnormality, the rest of the genome seems to be extremely stable. So when we actually design what base pairs we target this, we typically target about 20 base pairs at a time. And why we do this is if we can target 20 base pairs, we can try to find and design things that would have to have greater than 3 different mismatches that occur before it could possibly bind. And these different molecules are typically specific enough that that 3 base pair difference would cause it not to bind. But we're currently developing a lot of techniques and doing different techniques to actually evaluate the on-target profile, so when it actually binds and causes the effect in the region that we want it to be, and we can see how well it binds to that region, but we also try to look at all the off-target profile to see if it's actually going to bind anywhere else within the genome. So it's a really good question.
Thank you for answering my question.
You have a question, just raise your hand and I'll bring the mic over to you.
Could you go into a little bit more detail about how you're able to use, I think you're using PCAS9 to deliver the pCRISPR in the models that you were using. Am I correct in that?
Yes.
Okay. How are you managing to get the nanomolecules safely to those systems without the brain's immunization system actually killing those things off? That's kind of pretty interesting.
Right. So we're actually doing, we're starting those studies now to actually look at the immunological profile following injection. We actually haven't injected the dCas9 silencers into an organism yet. We're hopefully going to be doing that in the next couple of months. One of the ways that we think that we're able to actually address this problem is we're not actually injecting the Cas9 protein. We're actually doing encoding in our lipid nanoparticles, which should be able to kind of shield itself from any surface expression recognition from the immune system. Until it actually gets into the cell. And then once it's inside of the cell, that plasma DNA should transcribe into the protein. So hopefully we won't have any surface expression of the Cas9 protein itself outside. Right, and so the use of the lipid nanoparticles is a great method to actually to stably deliver DNA to a cell. But it also has several advantages, such as potentially avoiding recognition from the immune system. But all of that's an excellent question, and it's actually one of the major aims of our group moving forward is actually doing the toxicology and the pharmacokinetics of how these actual molecules get into the neuron, how the neuron responds to it, and then how the innate immune system responds to it as well. So it's an excellent question, and I think a lot of people in the field are at that juxtapose of trying to understand how these potent therapeutics will actually move forward from the immunological profile. Thank you for the question.
Thank you for the great talk. I was interested, so Diego asked a question and you were answering about how the transcriptional activator like enhancer specifically targets is it the CAG?
It actually doesn't target the CAG. We've made one, and we actually published a paper on this last year, well, I guess this year still, where we actually had one that would target the CAG expansion itself. And so in some of those videos, those regulatory switches that we see upstream of the gene, these regulatory domains, these regulatory elements, we actually try to land in those regions, because targeting the CAG repeat itself is problematic for several different reasons, both in the ability to be specific in targeting with the off-target profile, and then having the ability to actually try to shut down gene expression by targeting that exon. So we actually target just upstream of that in these regulatory domains.
Is there any way to collapse the array?
Yes. So we've done in one of our, in that publication that we were talking about, we actually created 2 different tall-based approaches to go to the CAG repeats and try to cause a truncation. We kind of stopped doing this because as we started to do some basic bioinformatical studies, in the human in most humans, you have 2 Huntington genes. And on average, we have about 15 to 17 CAG repeats. An actual paper just came out recently that there may be this large population of people that have the intermediate length, so this like 32 to 37. So not quite the threshold to cause Huntington's disease, but not low enough that kind of fits in the normal population. It's actually 1 in 400 people that have that. And so when you start to target, because of the size of the molecules and how we actually construct them, if we target 20 base pairs at a time and just in our different modeling techniques, we can be specific up to about 14 to 16 CAG repeats. And so if you had 17 CAG repeats, they would potentially target and cause a truncation. Anything over that threshold, it should cause a truncation. If you actually start looking throughout the genome, there's about 12 or 13 other genes that have greater than 18 CAG repeats. So even if you made something highly specific to anything 15 plus, 17 plus, 18 plus, it would specifically target that mutant Huntington, but by nature, it would have on-target effect to about 12 other genes. And so we've really moved forward with doing these epigenetic things, partially because they should also be reversible. If we actually truncate out part of the genome, it'd be very difficult to try to add that back in if something went wrong. Thanks. Yeah.
You kind of briefly mentioned how there was the limitation of translating these treatments to the human population.
Mm-hmm.
Could you kind of extrapolate a little bit on how the subfield, like what the dominant progression of treatment is going towards to translate this to human population, and a treatment that a lot of patients would say yes to?
Right. So I'll say the first thing that I think limits, and especially, again, based off of neuro, I showed you guys a really beautiful picture. That's, I think, one of Peter's favorite pictures that we have of that brain with half of the hemisphere completely lit up with this very potent therapeutic going in and potentially causing gene silencing. And we do volumetric. We actually look at how far it travels, how much it is, and we're really, really impressed with how much coverage we have. The human brain is about 10,000 times larger than the mouse brain. And so as these different therapeutics, traditional gene therapies with like AAV, cell transplantation, small molecule drugs, antisense oligonucleotides, those different things, in the mouse brain, you may be able to cover and hit the entire region that you're interested in. And for us, we were, I mean, again, blown away with how much expression that we saw. Those areas that we showed in those first sections where we saw those MRIs and we said, well, this blue area, this is deteriorating, that region is bigger than those regions collectively are bigger than the entire mouse brain. And so when we try to translate this, the biggest hurdle is the human brain's really big. And we would have to hit a lot of cells. We'd have to affect a lot of neurons. And there's probably a threshold in there about the percentage of neurons that you're able actually to create this in. Another major issue that I think we have to address is going to be the immune response, how we actually deliver these and how the body responds to it. In a post-mitotic neuron, these plasmids may exist for a very long time and produce proteins that are actually, and so the molecules themselves are proteins that are actually binding and causing this change. And we're talking about potentially having this protein being expressed inside of a neuron for maybe months to years, and we don't know what the response of the cell in the human body is going to be. And so we're limited in our models of how we study the brain and how we study neurological diseases. I can show you mice falling off of a rotating rod all day, and I can tell you that that correlates to motor coordination. You could talk to a patient that has Huntington's disease, and they'd say, I never ran on a rotating rod in my life. I don't know what that means. I can show you the anxiety things, and I can say, well, inside of this box, Huntington's might stay on the side, and that means anxiety. And a Huntington's patient could be in the room, and they would sit in the middle and not be anxious. So we have a lot of different things of how we translate both the size affecting the number of cells that we would have a functional consequence, and then also the models being able to extrapolate what we see in a mouse or a rat or even a non-human primate potentially someday, and how that's actually going to correlate to a human disease. Fortunately or unfortunately, Huntington's disease only happens in humans.
Thank you.
Hi again. How will you prevent this from being inherited still?
You wouldn't.
Okay. So each child will have to keep getting this treatment.
Right. So I think one of the most devastating things that we have going on, at least in the Huntington's realm, is you can know if you have the gene expansion. If you were a parent, you could know today, currently, you could go in and have your embryo tested, and you could see if it has that gene mutation or not. And you could actually learn very, very early on if that embryo that's forming, that eventual human that's going to be birthed and develop and live a normal life, if it's going to have Huntington's disease. There's all kinds of ethical considerations about when you can be tested. If you can get predictive testing, you have to be at least 18 and undergo genetic counseling and all these different types of things. You can get confirmational testing earlier in life. And so with this known, we know specifically what the gene is, and we know if the gene is going to cause the disease, and we can tell you very, very early in life. And it's a very cheap routine test to be able to do. Less than 5% of the people actually do it because they don't want to know. It's basically a death sentence at this point. And so what we were doing with this is trying to make it into a preventative therapy. There's a lot of people that live with pre-symptomatic or symptomatic Huntington's disease today, and we're trying to address those people. There's things like pre-implantation in vitro fertilization testing, where you could actually go in and have in vitro fertilization done. You could have several eggs get implanted and fertilized, and then you could go through and you could look at all of those eggs, and you'd say, out of these 30 eggs that we have, only give me the ones that don't have the mutation. 50% of the genes, or 50% of the embryos potentially. But that's an expensive process. That's not covered by insurance right now. No. In an embryo, like in a blastocyte? Yeah, I mean, well, if you had a surface expression marker to actually do flow, that would be the big thing. And because this is actually contained, most of the time the Huntington protein isn't an extracellular protein. You'll see it in aggregates forming actually within inside the cell. And so you'd actually have to lyse the cell to access this to know if the mutant protein is there. And then by the time you do that and then run it through a flow cytometer, those cells aren't going to be really viable anymore, right?
Well, that's the end of our series today. Can you please join me in thanking Dr. Fink again for joining us?